Okay, so yeah, so go ahead and make this, put Punnett square. You got a circle, which this represents a cell, right? And in this cell are a couple chromosomes shown. Again, understanding that there would be many more chromosomes. We wanted to look at all the chromosomes of a human. There'd be 46 of them, but we're just looking at one pair of chromosomes. We're looking at the trait of the earlobes, whether they're free or attached. And so what you see here, again, we can pick out, whatever, this would be some random female, right? And over here on this side is some random male. Okay. And so, again, the idea of this is when this female undergoes meiosis and makes eggs, what's going to happen to these two chromosomes? Yes. Are they both going to end up in an egg cell? No. They're going to segregate. Law of segregation. They're going to separate. And so I need to see if I can get a pen working here. All right, let's try this. So what you're going to end up with is, so you should do this, right? And we could draw the whole chromosome and all that, but we typically don't. So just understand, though, that when you're doing this on a Punnett square, you're actually showing meiosis. You're showing that these two alleles for the trait of earlobes in this female are going to separate from each other. Half of the egg cells that she makes are going to get the dominant allele. Half of them are going to get the recessive allele, because you would call this heterozygous. She has one of each type of allele. And then when you look at the man, same thing, right? He's heterozygous. He's got one of each allele. During meiosis, those are going to separate. Half the sperm cells he produces are going to get the dominant allele. Half of them are going to get the recessive allele. All right. And then what the Punnett square shows, so again, I'm sure you guys have done these. So what the Punnett square shows is all the possible chances of sperm and eggs. So if this sperm fertilizes this egg, you're going to get this child, right? So this is a possibility, right? You could have a child with both dominant alleles. Okay, if this sperm fertilizes that egg, you're going to get a heterozygous child. This is a possibility there. If this sperm down here fertilizes that egg, you're going to get a heterozygous child. And then if this sperm fertilizes that egg, you're going to get a homozygous recessive child, where both alleles are recessive, right? So there you go. Now, does this mean they're going to have 4 kids? No, it's just showing you what are the possibilities that these two parents could have. This doesn't mean they're having 4 kids. I have 1 kid. And in here, you could write down probabilities or whatever however you want to do it. So, but what's the chance of having homozygous dominant? 25%, right? So you want to put down 25%, you could put down 1/4. Okay. What's the chance? Again, these are all a 25% chance, right? Or 1/4. Yeah, fill that in like that. Okay, so there you go. Now, what you typically do then is you would kind of summarize what's going on here. So what I kind of sloppily have here in red is what's going on in these possible children. Let me get rid of all this green stuff here. Okay, so for genotypes, your chances are there, right? Homozygous dominant, that's how you call that. Please do not say big F, drive me insane. It's homozygous dominant. It's 25%. What do you call this one? Heterozygous, 50% chance. What do you call this one? Homozygous recessive, it's 25% chance. Now, if you're going like ACT or some tests like that, a lot of times they use, instead of percentages, they use ratios. So they might have this, 1 to 2 to 1, which means that for every one of these, there's 2 of these and 1 of these. So it's just the same way, just looking at it a little differently, say in ratios instead of percentages. Now for phenotypes, right, that and that, right, the homozygous dominant and heterozygous are both going to be free earlobes. So you'd say the chance of being free earlobe 75%, chance of being attached earlobe 25%, and if you were looking at that as a ratio, it would be 3 to 1 ratio. Remember? Yeah, it's a Dixon Ticonderoga. That's the best pencil made. Keeping it. Okay, there you go. That's how you do a Punnett square and how you'd analyze the results of it. And there you go. Okay. All right. Let's practice. Turn off my recorder.